Thank you for joining us today, and thank you to the Partnership for Clean Competition for their continued support and the opportunity to present my research. My name is Diana Velosa, and I am a second-year PhD student in the Chouinard Research Group at the Florida Institute of Technology in Melbourne, Florida. Today, I will discuss my ion mobility mass spectrometry research and its application to the anti-doping science. Ion mobility spectrometry, or IMS, is a gas phase technique that separates ions as they move through a buffer gas under the influence of an electric field. The simplest IMS is a drift tube shown here, which measures the time it takes for ions to traverse a separation cell with a static electric field. These ions undergo many collisions with the buffer gas molecules, and their drift velocity is dependent on their size, shape, and charge. This allows for molecular structure characterization and separation of compounds like isomers. Our Agilent 6560 IM QTOF can acquire an ion mobility spectra in under 60 milliseconds, which allows for convenient nesting of IMS within chromatographic separations and TOF mass analysis. This multidimensional separation workflow, LC-IMMS, provides the ability to analyze many WADA-prohibited anabolic steroids using extraction and isolation from human urine. This analysis yields three-dimensional datasets for each point on the chromatogram, where any given feature can be characterized by both drift time and mass-to-charge ratio. The ion mobility domain is especially beneficial in resolving isomers, which constitute a significant challenge not only for current-banned substances, but also for yet-to-be-determined compounds. Here are a few examples of isobaric and isomeric separations with the IMMS. First, fluoxymesterone and methylclostebol are isobars with the same nominal mass. Despite their similarity in mass, the compounds have different ion mobility drift times. We use this drift time to determine a compound's collision cross-section, CCS, a characteristic property of an ion under given experimental conditions. We are compiling a comprehensive library that catalogs CCS values for a range of performance-enhancing drugs, which can be useful identifier coupled with retention time and mass-to-charge ratio. Next, we have isomers boldenone and methyldianolone with identical exact mass of 309.18. However, they display different CCS, which allows for definitive identification. Lastly, we demonstrate the analysis of isomers epitestosterone and 1-testosterone, which also have identical mass of 311.2. Further complicating their identification is that our standard 10-minute LC-ROM provides very little separation between them. However, their different CCS allows for differentiation. This potentially provides an avenue by which to reduce or eliminate chromatographic separations, which will significantly improve throughput. To demonstrate quantitative LC-IMMS, we analyzed several concentrations of testosterone extracted from 1 ml human urine samples. This allowed for acquisition of the drift time versus the mass-to-charge plots in here. Traditional quantitative LCMS involves integrating peak area for an extracted ion chromatograph, filtered by an MSMS transition or accurate mass. This may not remove interferences due to chromatographic coelution or similar mass. However, filtering based on mobility dramatically reduces the noise. In this example, we have both known mass-to-charge ratio of 311.2 for testosterone and known collisional cross-section of 197.95 Armstrong square. This yielded double extracted ion chromatograms, filtered by mass and mobility over a wide range of concentrations. The results were quantitative over several orders of magnitude, with a limit of detection just below 1 nanogram per milliliter. Our current studies are focusing on improvement of extractions and fine-tuning instrumental parameters, which we believe will improve LOD and sensitivity. Aside from quantitative applications, ion mobility provides structural characterization and can allow the identification of novel substances. Here we study trends in mobility resulting from functional group differences. As an example, many WADA-prohibited steroids are simply testosterone analogs that differ only by a single functional group. For example, androgenic or nortestosterone lacks the C19 methyl group, highlighted here in red. Similarly, methylclostebol has an additional C17 group, highlighted here in green. The resulting spectra shows different collision of cross-section, with the least massive compound, mesterolone, displaying the smallest collision of cross-section at 194 nm². Although these compounds do differ in mass, such trends may allow us to identify isomeric species in the future. Furthermore, the methyl group contributes more to the change in mobility than would be predicted based on mass alone, as demonstrated by the reduced mass effect on the Mason-Champ equation used to calculate collision cross-section based on rev time. This brings us to my final slide, which outlines the computational approaches we incorporate into anti-doping studies. We have modeled many steroids and their complexes to determine structure and collision cross-section. The experimental and theoretical values typically match to within 1%. More recently, we have also incorporated a rapid, low-cost UV catalyzed reaction, including ozonolysis and the Paterno-Bucher reaction, to further provide structural information. As we piece together the many parts I have discussed today, we're finalizing a comprehensive workflow that incorporates experimental LC-IM-MS methods with computational modeling and machine learning algorithms to provide both targeted quantitation of known PEDs and simplified identification of compounds to be detected in the future. Thank you again for joining me in this opportunity to present my research. Please be on the lookout for the newest research coming from the Chouinard group.